Hi, everyone. My name is Chanel Hayson. I'm the Director of Outreach and Community Relations for the Elakha Alliance, and we're so pleased to welcome you today to our 4th annual Sea Otter Science Symposium. I'm going to pass it off to Bob Bailey, who is our board president for the Elakha Alliance, to introduce our next speaker.
Thank you very much, Chanel, and I'm going to share my screen, I hope, and get us to our next speaker, who is Dr. Leigh Torres. I had the pleasure of meeting her 4 or 5 years ago now at the early stages of this Elakha Alliance experiment that we're conducting here. And I'm thrilled she can be with us today because it's a great complement to the presentation we just heard from Dr. Rumrill. So Leigh received her master's in coastal environmental management from Duke University in Durham, North Carolina in 2001, and her Ph.D. in marine ecology and environment, also from Duke, in 2007. She is currently Associate Professor, Department of Fisheries and Wildlife and Conservation Sciences at Oregon State University, more specifically in the Geospatial Ecology of Marine Megafauna Laboratory. And you can look up that GEMM, the GEMM Lab, on the link that Chanel is going to put in the chat. They've got, it's a link to all kinds of fascinating information about what different students and faculty in that lab are working on. So the GEMM Lab focuses on ecology, behavior, health, and conservation of marine megafauna, including cetaceans, whales, pinnipeds, such as seals and sea lions, walruses, etc., seabirds, and sharks. Leigh and her students specialize in the spatial and behavioral ecology of marine megafauna, including marine mammals, seabirds, and sharks. And more specifically, she and her students have been conducting some really interesting work along the Oregon coast and at Port Orford around the use of kelp forests by gray whales. And I'm not going to spoil the show other than to say, welcome, Leigh, and thank you so much for being here today.
Yeah, thanks, Bob. Really awesome to be here. All right. Oh, you need to stop sharing, and then I will share.
Stop share.
Right. Okay, do the admin here. Okay, everybody can see and hear?
Yes.
Okay, great. Okay, well, yes, thanks for having me here today to speak to you about some of our work. Thanks for the intro, Bob. Yeah, so you might be wondering, well, why are we talking about whales when this is all about sea otters? And there's one really deep connection that we're finding, and that's through kelp. So that's what I'm going to tell you about today, is our sort of journey of discovery about the relationship between gray whales in Oregon, right off our nearshore coastlines here, and kelp. Before I dive in, I want to give a big shout out to my co-authors on this work, particularly Lisa Hildebrand, who is my PhD student, and then my postdoc Solène Derville and our collaborator Ines Hildebrand. All right. Okay. So Steve also mentioned these trophic cascades, but I'm also going to walk you through these concepts of trophic cascades because they're central to the work that I'll be telling you about today. So this is sort of the stereotypical kelp forest in the Pacific Northwest. You can see we have sea otters and sea stars, crabs, urchins, and kelp, as well as the drift kelp that sort of is the dead pieces that fall off kelp and to the sea floor. And that's really important because that feeds a lot of the benthic organisms. So the classic cascade is when sea otters are removed from the system. The loss of those sea otters release urchins from predation pressure, and then that causes those urchin populations to expand. And once they sort of expand too far that the amount of drift algae in the system won't sustain them, they end up directly eating the kelp itself. And the loss of that kelp then reverberates up through the ecosystem and can have impacts on other predators at the higher levels, such as crabs and octopus and fish. So these shifts between a really dense kelp ecosystem and these sort of urchin barrens have been documented for over 4 decades, and they're termed phase shifts, and they're not anything new. really, but the idea that they could turn into a stable state, so an alternative stable state moving from a kelp ecosystem to an urchin ecosystem, is what can concern us. And there's evidence from Northern California, our neighbors just to the south, that this might be happening. So this really good study and concerning study done by Rogers Bennett that was published in 2019 showed how after 2014, there was a dramatic loss in kelp. So that's what these boxes here show in the little green polygon. So these are 4 different sites that they surveyed in these different years. And you can see that after 2014, between 2008 and 2014, there was a dramatic decline in the amount of kelp at these 4 sites. And they think they make this strong case in the paper that this decline in kelp is due to 2 distinct events. One was the sea star wasting disease that began in about 2013. And sea stars, particularly Pycnopodia, are primary predators of urchins. So that can release those urchins from predation pressure and allow them to eat more of the kelp. And then the other event was the warm blob, this massive marine heat wave that took place in the North Pacific right off our coast here in 2014 through 2016-ish. And that warm blob was associated obviously with warm temperatures and low nutrients. And both of those factors, high temps and low nutrients, can really impact kelp growth. It's just not optimum conditions for kelp growth. So we have low kelp growth in those years and an increase in urchins. And that's likely what caused the shift from these rich, dense kelp ecosystems to no kelp. And you can see the transition in this figure from that paper, which shows this 90% loss of kelp across that ecosystem. And because the biogeography of this Northern California area is very similar to what we see along the Oregon coast, particularly Southern Oregon, there's a lot of similarities and anecdotal evidence. And now what Steve just presented earlier about this increase in urchins, we are concerned about similar phase shifts happening in our nearshore ecosystems. So if we return back to the classic Pacific Northwest sort of kelp forest ecosystem, we know, and that's why we're here today, in fact, that this isn't actually representative of our Oregon coastline because we have no sea otters here. They haven't been around for about a century. And so because of that, urchins in our ecosystems have really been under predation control by sea stars. But again, in 2013, we had that same massive sea star wasting disease, which removed them. And then we have that same warm blob episode, which caused low recruitment and growth of kelp. So we have also seen an increase in sea urchins in our along our Oregon coast. So now why are we in my lab that studies a lot of gray whales particularly interested in this? And that's because we've found through our research that the zooplankton, particularly these mysids, which are every single one of these little blips in this image here, they are highly associated with the kelp and reef ecosystems along our Oregon coastline. And this is the main prey item of gray whales that feed along our coast throughout the spring, summer, and fall months. So what we've hypothesized is that when there's high kelp, reefs with kelp will have more zooplankton, and then we'll have more gray whales feeding in those areas. But the contrast is also true, and that's that there could be indirect effects of increased urchins on gray whales through the pathway of reduced kelp, reduced mysid zooplankton, and then reduced gray whales. So we took an analysis to try and test to see if we could actually see these theories manifest in our data set. So, and just to give you some context for why we're actually concerned about the decline in kelp and zooplankton in this area, it comes back to the health of these animals. So one part of our research that we do a lot of is fly drones over these gray whales while they're here to look at their body condition. And what we do is we take a still image of the gray whale while it's flat and straight at the surface, and then we measure its body length. So this is whale length here. And then we measure its surface area between the 20% and 60% body length of the whale. And we calculate the body area index by dividing the surface area over the percentage of the body length of the whale. And this gives us a standardized metric of how fat, basically, the whale is. And we can compare that across individuals and individuals with different lengths. And basically what you need to know at this point is that BAI numbers that are lower are associated with skinny whales, and then BAIs that are higher are associated with fat whales. And there's a lot of data that I could show you about BAI, but what the basic message is that it does vary. The body condition of whales, so the BAI of the population is what we're looking at here, does vary across our years. You know, some years being quite high, but then it dips down and recovers. But what's interesting is that in the last couple of years, the BAI of our whales that we study here has been very low, and this has been concerning. So again, we think that there's probably multiple environmental factors that are influencing the prey availability to these gray whales, but we do think that the health of the kelp forests and therefore the zooplankton that's available to the whales to feed on is probably a primary factor that we need to look into. All right, so with that introduction, I'm going to transport all of you to beautiful Port Orford, Oregon, which hopefully many of you have visited. If not, I definitely encourage you to, because it's an amazing spot in the world that we're fortunate enough to run a long-term study site at. So we just completed our 8th year studying whales and zooplankton in this area. So this is our cliff-based study site where you can see one of our dedicated interns looking for whales. This is a theodolite, which I'll talk to you about more soon. But what we use this theodolite to track whales that feed in these nearshore areas. And so the whales feed in this area over here, Mill Rocks, as well as in this area here, Titchener Cove, that's just on the other side of the port there. So this is a map view of that area. So Port Orford is down here on the southern coast of Oregon. And this brown dot represents where that image was that I was just showing you, where a theodolite sits. And so from this dot, we monitor whales that move through our study area, which includes Mill Rocks and Titchener Cove. And within each one of these study areas, we have sampling stations, these blue boxes. And at the same time that we have a team monitoring whale movements from this cliff location, we launch a tandem kayak from a beach right here. And that team paddles out to each one of these stations daily and samples, it conducts GoPro drops and zooplankton net drops to assess the prey availability to the whales at the same time that we're tracking their movements. Right, okay, so there's, yes, theodolite tracking of the whales, and there's our prey assessments using GoPro drops and nets. So here's the team in action with a theodolite looking for whales. So a theodolite, if you're not familiar, it's a very old, actually, tool that surveyors use to map the landscape. But essentially, it works by knowing the exact elevation of your theodolite, and then you get fixes on whatever point you want. In our case, it's where the whale is. And that theodolite calculates the angle. So based on those 2 bits of information, you can get fixes about where the whale is exactly in the world. And this is an example output from one of those tracks of whales. So again, we have our coastline of Port Orford here, this dark black line, our cliff site monitoring location, and then our Mill Rock study area and our Titchener Cove study area. And what this black line is here with all the little circles, each one of those circles is a fix that we made of where the whale was moving through this habitat. And once you string each one of those fixes, those little circles together, you get a good picture of the whale's movements. And so you can see here that the whale, we started tracking the whale here, and it moved through this area, came into Mill Rocks, did some circling about, likely feeding or searching in this area, and then it transited over here, did a loop-de-loop, searched for something, then came over here to the port, also did a loop-de-loop looking for something, probably didn't find what it was looking for, and then transited right through Titchener Cove on its way out. So we've collected many of these tracks of whales over our past 8 years of fieldwork. At the same time, this is what we do in the kayak. We drop the Secchi disc to look at water clarity off the back of our kayak. And then we drop a GoPro stick, which takes images of the water column, so we can calculate the amount of zooplankton abundance. Then we drop that net, and from that we get a sample of prey. And later in the lab, our dedicated team of interns and graduate students counts and enumerates each one of those little zooplanktons that we catch. All right, so what I want to focus on is those GoPro images. So these are some images from one of our sites in Titchener Cove in 2016. So as you can see, there was a lot of kelp. You can barely see anything else. But what we noticed is that at that same location in 2018, it looked like this. And this was quite startling to us. There was very little kelp, just these sort of mowed on stalks. You can see urchins here crawling on the stalks. And then sort of barren reef with a lot of urchins all over it. So this was concerning to us, and we decided to see if we could come up with a way to analyze these data in a robust manner to look at patterns of change in kelp and urchins and zooplankton. So what we did was, for each image that we extracted from each station across our years and study sites, we calculated sea urchin density, which is the number of urchins we could see divided by the number of urchins that could have been in that image. So based on available reef substrate or kelp stock area. Then we qualitatively assessed the kelp health in each image in a standardized way. So there were, oh gosh, what were the metrics? It was like very healthy, poor health, or unhealthy. Actually, I can't remember at the moment, so I shouldn't even say, but we converted it to a numerical score for the analysis. And then we calculated the relative abundance on each GoPro cast. So the whole cast, we calculated the abundance of zooplankton in the images. And then, of course, we also calculated the amount of foraging effort by gray whales at those stations, so within 30 meters of each one of those stations. So I'm now going to jump into the results to give you a sense of what we saw. So I'm going to show you a bunch of little panels like this. So I first want to orient you. So this gray area is the shoreline of Port Orford. Again, these are our study sites, Titchener Cove and Mill Rocks. Each one of these circles represents one of our sampling stations in that year, with the average amount, in this case, of sea urchins. So if it's a light color, there's low sea urchins. And if there's a darker color, there's higher sea urchins. So what you can see here is that in 2016, for sea urchins, we had very few in Mill Rocks, and in Titchener Cove, we had just a little bit at this one site. So now if I add in the other taxa that we're looking at in this study, you'll see that kelp in the dark green, you can see we had a lot of kelp in Titchener Cove. It was very healthy, and a good amount out in Mill Rocks as well. For zooplankton, we found a lot of zooplankton, so dark orange in Titchener Cove, and some as well out in Mill Rocks. And then for gray whale foraging effort, we had a good amount in both of our study sites, particularly here in Titchener Cove and in these locations in Mill Rocks. So now let's add in the rest of the years for sea urchins. And what you can see is a dramatic change. And that is that where we had lighter circles, it now goes to dark purple, which means high density of sea urchins at both of our sites. So you can see that happens at Titchener Cove, a transition, as well as a transition in Mill Rocks. And likewise, we see the opposite for kelp. We see basically a fading from dark green to lighter green across our time in both of our sites. But what's interesting to notice is that we started seeing this change first in Titchener Cove, right? That sort of declines more quickly. And then it's sort of a wave, we call it. Then we started to see that transition to less kelp health in Mill Rocks as well. And we see a likewise change in the zooplankton relative abundance across our study time as well, moving from sort of dark orange to very light orange. By our final year, we basically had almost no zooplankton caught in our nets or on the GoPro. And likewise, as you'd expect, we certainly transitioned from higher foraging effort by gray whales to very low, a presence even of gray whales in the ecosystem, and very little foraging at our study sites as well. So that gives you sort of a visual representation of the data that we collected, but we also did statistical analyses on these data. So this is moving into the statistics part, but this is where you get your quantitative numbers to what you visually see. And we used two methods to assess the relationship between these four taxa. One is just a simple Pearson's correlation. That's how related numbers are to one another, but it doesn't account for any spatial structure or temporal structure. So we have spatial structure in our data because we have sampling stations at different sampling stations across our study area. And then we also have temporal structure in our data because we sampled across 6 different years. So we used a method called joint species distribution models, and we inputted our data into that. So I'll walk you through our results now. So first I'll show you this fun little diagram that Lisa made, my graduate student, but it shows our 4 taxa that we're interested in: gray whales, urchins, kelp, and mysid. And these lines, which are coded by whether they're a direct effect or indirect effect, are connecting our different 4 taxa. So you can see there's a direct effect between urchins and kelp because they eat the kelp, as well as a direct effect of whales on mysids because they eat the mysids. And then we have indirect effects between the other components of our study system. So with the Pearson's relationships, and this is where we just group all the data together without considering the effects of space or time, we see what we expect, although some weak relationships, and that's negative relationships between urchins and whales. I should say that the blue numbers represent a negative relationship and the red numbers represent a positive relationship. And they will scale between 0 and 1. And 0 means very little relationship, and the closer the number is to 1, it's a very strong relationship. So you can see here that the strongest relationship we see is a negative relationship between between urchins and kelp. So, but now if we move into the results from the joint species distribution models, where we're only accounting for space, we start to see some interesting results. And this is that urchins still have a negative relationship with the other taxa. So that would be whales here, as well as the mysids, and then a stronger negative relationship with the urchins. And then we see, but what's interesting is that this relationship between whales and kelp is positive. We have a 0.28, so it's trending towards a higher positive relationship. And that is indicative of what we have seen in some of our other results, that gray whales really do prefer habitats with kelp. But what's interesting is that this relationship here, that gray whales relationship with mysids, is actually turning into a negative relationship, as is the relationship between mysids and kelp. So this is not what we expected, which was interesting. And then when we add in the temporal component, so now we're accounting for both the spatial structure and the temporal change across our study. What we see is that there are very strong negative relationships between urchins and whales, so negative 0.97, which basically indicates that whales avoid habitats with urchins, or really don't like those habitats. But we see a very strong relationship between whales and kelp, so whales definitely like kelp habitats. We also see a very negative relationship between the urchins and the kelp. So that shows what we all know is that the urchins directly impact kelp habitats. Where you have a lot of urchins, we have very little kelp. But what's interesting is to see that we still have this very strong negative relationship, or not very strong, moderately strong negative relationship between whales and urchins, as well as kelp and whales and zooplankton, which is not what we'd expect. We would think that the whales would go with theirs prey. But there's also this really interesting negative relationship between the zooplankton and the mysids. So while this was a bit confounding to us, what we think is that there's some other factor going on that's influencing where zooplankton aggregate. And so this definitely needs more research. So it's not just about bull kelp and where bull kelp is. There's something else that manages to aggregate the zooplankton. So, but when you look at the model outputs here from the same model, from the same spatial temporal joint species distribution model, we see, sorry about that, we see these really clear relationships that as urchins dramatically increased over our sampling period from 2016 to 2021, we see dramatic declines at the same rate between kelp, zooplankton, and gray whales. So again, this relationship, these patterns of zooplankton are really interesting to us. And we do think that what our working hypothesis is that even though zooplankton has become less abundant over time within our study system, they might become more spatially clustered in those areas where there is good habitat for them. So this obviously needs more research to understand what's driving these patterns of zooplankton, because they are such a critical component of the trophic food web in our nearshore Oregon habitats. You know, they supply not just food for gray whales, but many different species of fish as well, recreational and commercial fish. So with that, let's revisit our cascade where urchins have been released from their only predators, you know, in our study system anyway, that would be sea stars. And what we can do is add in gray whales and mysid shrimp to this trophic cascade. And what our study is the first one to document that there are correlative effects of this sort of traditional thought perception of this trophic cascade on mysid shrimp and then on these top predators of gray whales, which we have documented are now declining, or at least up until 2021. So just in summary, what I want to say is that while we've documented these interesting effects, you know, between 2016 and 2021, a lot in Port Orford, we're not really sure if this is a phase shift or, you know, into this alternative stable state, or if it's just, you know, a more natural oscillation, pendulum swinging back and forth. I was interested in Steve's talk that he just gave. He pointed out that there was this massive sea urchin recruitment event in 2014 and 2015, which aligns when we started seeing our increased urchins in our little study system. So obviously that aligns, and it's yet to be seen how these urchins respond. And maybe as our whole ecosystem in the Oregon coast recovers from the warm blob, kelp might be able to respond and be resilient in these important reef ecosystems. So just in summary, while, you know, just to bring it back to the gray whales, I guess, and why we care about this is that the body condition of gray whales is a really good metric and indicator of not just their health, but the health of the ecosystems on which they depend, which happens to be the same ecosystem of which sea otters will likely depend if they are reintroduced to Oregon. So as my last slide, I just bring in this, this figure that comes from a paper that my student Dom Kone produced that was published in 2021. That predicted where would be high sea otter densities based on habitat availability. And what I just want to point out is that one of the primary areas with high red colors, which means high density, is this area in Port Orford. And what I've been wondering as we go forward with the discussion about sea otter reintroduction and how and where might be good habitat is perhaps the distribution and foraging habitat use of gray whales could be used as an indicator of what could be good habitat for sea otters, because they're probably both looking for healthy kelp ecosystems to feed in. So that's all I have for the moment. And yeah, I think there's probably some time for discussion or questions, and I'll pass it on from there.
Thank you, Leigh. That was amazing. If you have any questions, please use the Q&A feature to submit them. I just want to say for the first time this year, I saw gray whales on the Oregon coast in Depot Bay, and it was like one of the most amazing experiences. because luckily the weather was in our favor, and it was hot and no wind, and the ocean was calm, and we were just following, I think her name was Gretchen. She is a common whale seen out of Depot Bay all the time, but she was doing the sharking behavior outside of the kelp forest, and it was just fantastic. So thank you for sharing all of that information with us. We don't have any questions. Oh, here we go. I'm like, where is everybody? Let's see. Grant asks, does the whale's low BAI have anything to do with the high population of gray whales around 2015? Possible starvation from a high population alone?
The high counts in 2015. Is he referring to the PCFG, like the whales in Oregon, or does because overall, I'll back up a bit, I guess. So there's the large Eastern North Pacific population of gray whales, which is estimated to be about 20,000, but it definitely oscillates up and down for various reasons. But most of that population, 99% or more, migrate up to the Alaskan Arctic area to feed during the summer. So it's this very small group, about 230 individuals that don't make that full migration. And they're called the Pacific Coast Feeding Group. And they're the ones that feed between Northern California and British Columbia each summer. And those are the ones that we study here. So so I think probably what this questioner is asking about is about the unusual mortality event that's going on with gray whales. It was started in 2019, and it actually is continuing on. And what's interesting there is, we did a study using drones. We compared the data that we have of the body condition of gray whales here in Oregon with the body condition of these ENP whales. They're called Eastern North Pacific whales, which basically means all the whales that aren't this PCFG. And what we did was we compared the body condition over the same time period, so 2017, 18, 19, leading up to the UME, to see if these two different populations have the same response. And what's interesting is that while the population of the ENP whales, that body condition definitely declined dramatically between that period, the PCFG whale body condition actually increased up to that point. And so we think that there's different environmental factors at play controlling the different prey in these two habitats. So up in the Arctic, there's some extreme changes going on associated with climate change. And then here, I think what we've seen is that the ecosystem actually recovered after the warm blob. And so while the body condition of gray whales here declined from about 2016 and 17, after 2017, as the ecosystem recovered, their body condition also recovered. So I hope that kind of answered the question in a roundabout way.
Okay, we have a couple more questions for you. Do gray whales feed in bays and estuaries?
No, not traditionally, but there is an exception. There's a group called the Sounders that is really only 12 whales that go into Puget Sound, and there they feed on ghost shrimp in mudflaps for about 4 or 5 weeks every sort of spring. They're actually this really interesting group of whales that they're not part of the PCFG. They're actually that ENP group that goes all the way up to Alaska, but they found this little interesting strategy where they duck in and feed, and then they continue north. But traditionally, no, they don't feed in estuaries. Every now and then, I mean, gray whales are really interesting because I think they're risk takers is what I call them. They're sort of very plastic in their feeding habits. They're not stuck on one thing. If they don't find what they're looking for, they'll be like, oh, okay, I'll go up this river and see what's up there. So every now and then you hear about a whale going up a river or in an estuary. And I think during that UME in 2019 or 2020, there were 6 or 10 gray whales in San Francisco Bay feeding. But they were incredibly emaciated and sick. So clearly they were, desperate and trying to feed on stuff.
Thank you. Dave asked, what % of their food do you think they get from plankton?
Well, they certainly indirectly feed on plankton. Do you mean, sorry, phytoplankton? Like, because so right, so there's two different sort of groups of plankton, right? We have our phytoplankton, which are like the plant life, the ones that depend on chlorophyll. And then we have our zooplankton, which are more, you know, meat eaters. They eat other things.
They said zooplankton.
Okay, zooplankton. So yeah. Oh, as opposed to maybe like amphipods and things like that, because I think their major diet here in Oregon is mysids. They also feed on amphipods, which are little, also zooplankton, but they're more in the benthos. So we do see the whales here feeding with many different tactics. So sometimes they are sort of benthic feeding, where you can see like massive amounts of mud streaming out of their mouth as they filter it out, but other times they're feeding on this zooplankton associated reef. So not really on the benthos, but just right next to the reef, gulping it up. Sometimes we've seen them feeding on surface aggregations of crab larvae, so particularly porcelain crab larvae. So they really can change it up based on what prey is available for them.
Thank you. Let's see. What would you predict is happening with gray whales in the areas that Sarah Hamilton, who's doing kelp research, showed were not experiencing a kelp decline?
So my understanding from Sarah's work is that she did those studies in the offshore reef. I assume we're talking about what she did for her PhD. And so those offshore reefs are really interesting. They're not where gray whales feed. So I think one of her study sites was Orford Reef. So we're just off the shore of Port Orford. And we've gone out there to survey and look for whales. And we have not seen any whales there ever, which is in contrast to a place like Cape Blanco, which is very close, or Port Orford, where we do find a lot of whales. So not really sure why, but either it's because, you know, obviously there's not food there would be why the whales aren't there, but why there isn't food there, I'm not entirely sure. Maybe it's deeper habitat or not the right nutrient load for them. Actually, that would be a good question for Steve, to see if they see the same amount of mysids out in that Orford Reef area as they do in the more nearshore areas. Yeah, because one other I thought is I know they have a lot of sharks out in the Orford area, so I always wondered whether that was a driver of things as well.
Yeah. Oh, is that Steve? Oh no, it's Bob. Okay, we have one more question right now. Why do zooplankton like the kelp forest ecosystem so much?
Yeah, great question. I wish I knew the exact answer, but well, we have a few theories. I can tell you the theories. One is that the kelp aggregates nutrients. Basically, it acts as, you know, this retention area where nutrients, you know, aggregate there. One other reason is that it reduces current flow. So there's not so much water movement, and the zooplankton can aggregate there more easily. So those are our two sort of primary theories that we're working on. But I have another student, Allison Dawn, her whole master's thesis is basically focusing on zooplankton and trying to figure out what the drivers are of their nearshore abundance. It's really interesting. We know a fair bit, a good amount of what drives zooplankton abundance and diversity in the offshore ecosystem of Oregon, so the whole upwelling complex. But we know very little actually about what happens in these nearshore environments, you know, around these reefs and headlands. And that's because it's a very hard place to work and study because of swell and the rocks and kelp and everything. So yeah, which is why we use some of these creative methods like GoPro drops and kayaks and things, because they're a little bit more nimble in these habitats. But yeah, lots of really interesting questions. So Allison's work is trying to relate our zooplankton catch data to temperature and productivity, but as well as winds and tides and dissolved oxygen. And then retention, so distance from headlands. So we wonder whether sort of the headlands in these nearshore environments actually retain nutrients, and that's why, and that might be actually why Orford Reef, because it's not near a headland. So maybe areas that are sort of protected by headlands are these retention areas for nutrients. So at the moment, there's a lot of theories, but we hope to get there in the next few years.
Wonderful. Well,
I,
that, go ahead, Bob.
I call the retention effect of kelp forest the dust bunny effect.
Yeah, good one.
Likes of your chairs and your tables, that's where the dust bunnies collect for sort of the same reasons. That's my simple analogy.
Yeah, Leigh, I'll use that. Thank you.
No problem. Trademark and all.
Yeah.
Talk about other whales and whether or not any of the other great whale species that along our coast, how they play into any of this, or are they too far offshore?
Yeah, well, gray whales are really the only ones that feed really this close to shore. Yeah, certainly off our coast, we have blue whales and humpbacks and fin whales and lots of other, like a fair amount of diversity, sperm whales, killer whales. But they're, much further offshore. One thing we're also really interested in, and so this is another part of my work, is that we collect the poop samples, fecal samples of gray whales, and we do that to look at their hormones as well as what they're eating and the microplastic loads in that they're ingesting. But there's this thing called the whale nutrient pump, which I don't know if you're all familiar with, but basically it's this idea that whales fertilize the ocean, because and they do that both vertically and horizontally. So whales, feed at depth, and then they come up to the surface and they'll poop. And then within that poop is a lot of nutrients that fertilize the ocean. And they can also do that horizontally too, meaning that they might feed over here and then travel over here and then poop there. So they can be a big distributor of nutrients across our oceans. Our oceans are really nutrient limited, which is why they're, for as vast and big as our oceans are, there's really these pockets where there's the productivity. So one of the main pockets is, the California current system. That's a very high productivity area, but most parts of the ocean are not. And that's because they're nutrient limited. And so whales are really important parts of our ecosystem, not just because we like them and they're cool or whatever, but because they actually fertilize the ocean. So, gray whales. So one thing I do want to do eventually is calculate the amount of nutrients and carbon contributions that whales provide, gray whales, to our reef ecosystems here, because that would be a double whammy if gray whales are leaving our kelp ecosystem because there's not food there, but then they're also not there to supply it with nutrients to help it regenerate. So in the offshore ecosystem, that's probably also happening too, with what humpback whales, defecating, and blue whales there as well.
Certainly, Ralph Chami, and our keynote speaker, spoke about the whale pump effect.
Oh, okay, great.
Contributing to overall natural capital of the world's oceans.
Indeed, yeah.
Well, let's see. Any more questions, Chanel?
No, we don't. But I'm just going to throw in here the prize for the end of this segment, randomly chosen. I've got another beautiful sea otter print and some kelp for Josie Islin. Hi, we're on this whole time. Congratulations. Thanks for watching and participating. I will mail this off to you. But yeah, so I think we are finished with our first half of the symposium. We're going to give you about an hour and a half of a break and come back here at 1:30 for the rest of our fantastic presenters.
Yes, there will be a lot of related topics this afternoon that tie right back into Steve's talk, to Leigh's talk, and to Ralph's talk for that matter too. So looking forward to it, and we'll see you at 1:30 Pacific Daylight Time. Thank you, everybody.